Hi everyone. Today's topic is anterior pituitary hormone. Synopsis, introduction, physiological anatomy of pituitary hormone, synthesis of growth hormone, mechanism of action of growth hormone, physiological actions of growth hormone, and the applied physiology. Introduction. The hypothalamic pituitary unit forms a unique and important component of the entire endocrine system that regulates the growth, lactation, fluid homeostasis, and the functions of thyroid, adrenal glands, and the gonads. Functional anatomy. Pituitary gland, which is also called as hypophysis cerebri, is a small gland which weighs about 0.5 grams and is approximately only 1 centimeter in diameter. It is situated in the hypophyseal fossa, that is the sella tunica of the sphenoidal bone. So this is suspended by a pituitary stalk which connects it with the hypothalamus. Development. Anterior pituitary hormone is ectodermal in origin. It develops from the Rethke's pouch, which is an embryonic upward outpouching from the roof of the primitive oral cavity. Posterior pituitary or the neurohypophysis, it develops from a lowered outpouching of the neuroectodermal tissue from central areas of the hypothalamus. So it is clear that the anterior and the posterior pituitary develops independently from two different areas, and it is only a coincidence that when they formed they are lying together very close, so they are considered the parts of the same organ, but their origin and their source of origin is completely different. Parts of the pituitary gland. Physiologically, the pituitary gland consists of three distinct parts or the lobes. So anterior lobe or adenohypophysis, posterior lobe or neurohypophysis, and then the intermediate lobe or the pars intermedia. Adenohypophysis, the glandular anterior lobe of the pituitary gland, is called as the adenohypophysis. It constitutes about 80% of the pituitary gland. It can be further divided into three parts, that is the pars distalis, pars intermedia, and pars tuberalis. So pars distalis, you can see it here. This is the pars distalis. It's the main bulk which is highly vascular, and pars media, it is in the pars intermedia, it is in the middle, so it is a avascular zone. And then pars tuberalis, here it is the pars tuberalis. This is the most vascular zone and it contains the secretory cells. Neurohypophysis, the posterior lobe of the pituitary, it is a neural structure and hence it is called as neurohypophysis. It consists of three parts. Pars posterior, it is also called as the infundibular process, which forms the main bulk of the neurohypophysis. Next is infundibular stem. It is the funnel shaped extension arising from the median eminence at the floor of the third ventricle, and the third part is the median eminence. So it is a small protrusion from the base of the hypothalamus. It is situated under the third ventricle, and it is highly vascular. Pituitary stall, the median eminence and the infundibulum that forms the neural stalk. So both together. It is surrounded by pars tubralis of the adenohypophysis. Intermediate lobe of pituitary gland is rudimentary in humans as well as in a few other mammalian species. In certain lower animals, this lobe secretes melanocyte stimulating hormone in response to changes in exposure to light and other environmental factors. In humans, it contains very few melanocyte containing melanin granules. ACTH has a very weak melanocyte stimulating hormone like activity. So in case of hypersecretion of the ACTH, there is hyperpigmentation of the skin. Histological structure of the pituitary gland, adenohypophysis, the pars distalis consists of cords of cells separated by fenestrated sinusoids. The cells can be divided into two main types, that is the chromophores and the chromophils. So chromophores, these are A granule cells, and it is considered that chromophils are derived from the chromophores. Chromophils or granule cells constitute 50% of the cells of the anterior pituitary. So these chromophils are further classified as acidophils and basophils. So these cells secrete growth hormone or the somatotropin, prolactin or the lactogenic hormone, and then the basophilic cells, these granules are basophilic and they secrete ACTH, beta lipoprotein, and also thyrotropic hormone, which stimulates the activity of the thyroid gland, and also they secrete follicular stimulating hormone and luteinizing hormone, and finally folliculostellate cells. These cells send processes between the secretory cells. Recently it has been found that these cells contain and they secrete the cytokine interleukin 6, but their physiological role is still not clear, and there is still few researches going on in it. Histologically, posterior pituitary contains three structures. One is unmyelinated nerve fibers. These are the axons of neurons located in the supraoptic and paraventricular nuclei of the hypothalamus. They carry the precursor of posterior pituitary hormones, and they end as closed terminals near the blood capillaries. Second one is pituocytes. They are the special cells, special type of the supporting cells having long dendrites dendritic processes. These are present between the axons. And finally the glial cells. So they are like astrocytes and the oligodendrocytes also seen. This is also blood supply. The arterial blood supply is mainly by the branches of internal carotid arteries, which forms the superior and the inferior hypophyseal branches, and then anterior cerebral artery and posterior cerebral artery. The blood supply from the anterior pituitary that drains into the cavernous sinus and finally into the jugular vein. So you can see that the anterior pituitary lies outside the blood-brain barrier, hence it is accessible to influence from the general circulation. So it can influence it can stimulate the secretion. So hormones and the neurotransmitter by the brain and hormones secreted in the general circulation can change the actions of the pituitary hormone. Hypothalamo-hypophyseal portal system. The branches from the superior hypophyseal artery form a ring around the upper part of the pituitary stall, and the branch and they are further branched to form a capillary network. The blood from this capillary network is drained into a long portal vein in the infundibulum. Then in the anterior lobe, these long portal veins break into another set of capillary network and represent as the sinusoids of pars anterior. So this arrangement is called as a hypothalamo-hypophyseal portal system. The inferior hypophyseal branches, the inferior hypophyseal network, which is also a branch of the internal carotid artery, they branch out and they form a capillary network at the lower end of the infundibulum stem. The short, you can see this is the inferior hypophyseal artery, which forms a capillary unit, and this is the short portal vein. The short portal vein, it arises from this capillary network and supply the blood mainly to the posterior pituitary and some parts of the anterior pituitary. The short portal vessels provide the link between the anterior and the posterior pituitary. Here it is. And this is the vein. The influences from the hypothalamus are conveyed to the pituitary gland by two different tracts. One is the hypothalamo-hypophyseal tract and tubero-infundibulum tract and hypothalamo-hypophyseal portal system. Hypothalamo-hypophyseal tract, it is composed of axons of large neurosecretory cells of the supraoptic and paraventricular nuclei of the hypothalamus. These fibers pass through the neurohypophysis through the infundibular stem and form a series of dilated terminals, which is called as the Herring bodies. Herring bodies. The neurosecretory cells of the supraoptic and the paraventricular nuclei, they secrete peptide hormones and which is the vasopressin and the oxytocin. They travel down their axons and in the neurosecretory granules, which is to be stored in the nerve terminals lying in the neurohypophysis. So based on the stimulation of the cell bodies, the granules are released by the process of exocytosis, which is released through the inferior hypophyseal artery. Tuberoinfundibular tract and hypothalamo-hypophyseal portal system. So the cell bodies of the hypothalamic neurons synthesize certain releasing and inhibiting hormones, which are conveyed by the infundibular tract to the median eminence regions, where they are stored in the nerve terminals. After these, the hypothalamic neurons are stimulated by the nerve impulses. So the releasing or the inhibition of the hormone are discharged into the median eminence, and they enter the capillary plexus of the superior hypophyseal artery. From there, they are transported to the portal vessels. So that is long portal veins, and then they exit from the secondary capillary plexus to reach the target cells in the adenohypophysis, where they regulate the secretion of the tropic hormones of the pituitary, anterior pituitary. The main topic is the anterior pituitary hormones. The anterior pituitary hormone is truly the master endocrine organ. The hormones are broadly classified into three categories: the growth hormone family, glycoprotein hormone family, and pro-opiomelanocortin peptide family. Hormones of growth hormone family includes growth hormone and prolactin, which are secreted from the anterior pituitary. And human chorionic somatomedinC, they belong to this family, but it is secreted from the placenta. Glycoprotein hormone family, the hormones of this family secreted by the anterior pituitary hormone. It includes thyroid stimulating hormone, luteinizing hormone, and follicular stimulating hormone. Pro-opiomelanocortin peptide family. The hormones are derived from a single precursor, the pro-opiomelanocortin. It consists of 241 amino acids. The precursor molecule is then cleaved into three fragments, which include adenocorticotropin hormone, melanocyte stimulating hormone, that is alpha and beta, which is produced in the intermediary lobe, and then beta lipotropin and beta endorphin. The physiological aspects of the growth hormone is going to be discussed in this class. So other hormones will be discussed later. So the TSH along with the thyroid hormones, the ACTH along with the adrenal gland. And in the next class we will see about the prolactin. So growth hormone, it is secreted from the somatotrophs that constitute about 50% of the total endocrine cells of the anterior pituitary. As the growth hormone promotes the postnatal somatic growth, it is also known as the somatotropin. It does not stimulate the fetal growth. It maintains the normal lean body and bone mass in the adults. It has many metabolic effects. It has the structural resemblance with the prolactin, and also the placental lactogen. Human growth hormone is a polypeptide which contains 191 amino acids. There are two forms of growth hormone. One is which has the molecular weight of 22,000, that constitutes around 90% of the growth hormone. The second one is which has a molecular weight of 20,000, that constitutes around 10% of the total circulating growth hormone. It is in the long arm of chromosome 17, contains the growth hormone, human, chorionic, somatomamotropin cluster genes. So humans, they exhibit a species specificity to the growth hormone. So it has diurnal variation which is secreted episodically. Synthesis: growth hormones is synthesized as a large prohormone in the rough endoplasmic reticulum. The prohormone consists of N-terminal signal peptide and 191 amino acid peptide hormone. Subsequently, the signal peptide is removed when the prohormone reverses through the Golgi apparatus. Finally, the hormone is packed and stored in the granules of the somatotrophs. Regulation of the growth hormone synthesis. Two hypothalamic hormones control the synthesis of growth hormone. One is the growth hormone releasing hormone and the somatostatin. So the GHRH, they stimulate, and somatostatin, that inhibits the synthesis of the growth hormone. The growth hormone regulating hormone promotes the growth hormone synthesis by stimulating the expression of growth hormone genes in the somatotrophs. TSH, that also stimulates the expression of the growth hormone gene. Therefore, patients with the thyroid hormone gene deficiency, they also develop the growth hormone deficiency. Regulation of growth hormone secretion. The normal basal plasma growth hormone concentration, it ranges from 0 to 5 nanogram per milliliter in the adults. The rate of the growth hormone secretion, it primarily depends on the balance between the action of GHRH and the somatostatin on the somatotrophs of the anterior pituitary. The GHRH stimulates and the somatostatin inhibits the growth hormone secretion. The growth hormone in turn, it controls its own secretion by the feedback mechanisms. The growth hormone has a negative feedback effect on the GHRH and a positive feedback effect on the somatostatin secretion. The growth hormone stimulates the production of insulin-like growth factor 1, or it is also called as SomatomedinC, which in turn has a negative feedback effect on the growth hormone secretion. The insulin like growth factor 1 has effects on both at pituitary and the hypothalamic level. There are various factors that regulates the growth hormone secretion by influencing the secretion of the GHRH, somatostatin, and the insulin-like growth factor. So recently, a new growth hormone releasing factor is found, and that is secreted from the hypothalamus, which profoundly they modulate the growth hormone secretion. That is the ghrelin, Ghrelin is proposed to be the new growth hormone releasing factor. There are various factors that regulate the growth hormone secretion. The main influence comes from the secretion of the GHRH, somatostatin, and the insulin-like growth factor 1. The other factors that increases the growth hormone secretions are hypoglycemia, as occurs in exercise and fasting, and then hyperaminoacidemia, as it occurs after a high protein meal, and then hormones like estrogens, androgens, acetylcholine, serotonin, and glucagon that increases the growth hormone secretion. Then stress during fever, surgery, or any psychological trauma during puberty, which is very essential. And in stage 4, sleep. And then there are drugs that increases the growth hormone secretion like levodopa, alpha receptor agonist, and apomorphin, and enkephalins. And the factors that decreases the secretion of the growth hormones or when there is increase in the glucose and free fatty acids, and hormones like cortisol, growth hormone itself, and medroxyprogesterone, they decrease the growth hormone secretion. And then during REM sleep, obesity, pregnancy, all they decrease the growth hormone secretion. Growth hormone secretion throughout the life. So growth hormone secretion increases during the infancy and is maintained throughout the childhood and increases enormously at puberty, which is associated with the sudden increase in height and general growth of the body. So after puberty, the growth hormone secretion decreases to the adult level and it maintains still the senescence. So the final height of the individual is mainly determined by their inherent growth hormone secreting capacity. Growth hormone secretion in sleep, it is not clearly known why growth hormone secretion occurs in episodes or bursts throughout the day when the growth occurs slowly over months and years. The pulsatile secretion becomes more prominent in the midnight, especially within few hours of the deep sleep, approximately 70% during the slow wave sleep in stage 3 and 4. Individuals who sleep adequately, they usually remain healthy. That is the reason we ask all the people to sleep well. Children, they are advised to sleep more to attain normal growth, as this is the growing phase of the life. That is why the infants, the newborns, they sleep a lot. It helps them to secrete more growth hormone and grow normally. Metabolism of the growth hormone. The growth hormone binds with the plasma protein, which is a growth hormone receptor fragment. Growth hormone receptor fragment, which is a concentration of the plasma protein, is the index of the number of growth hormone receptors present in the tissue. So about 50% of the growth hormone is bound to this plasma protein, the reservoir. Growth hormone is metabolized rapidly by the liver. So the half-life of the growth hormone is about 6 to 20 minutes. Normally in adults, less than 3 nanogram per milliliter of growth hormone is present in the plasma in the basal conditions. Though only a small quantity of the growth hormone is excreted unaltered in the urine, the daily urinary output of the growth hormone reflects its 24 hours profile. Mechanism of action. The growth hormone binds with the receptor called growth hormone receptor, which is a tyrosine kinase associated receptor that belongs to the member of cytokine receptor superfamily. So it has three domains: a large extracellular domain and transmembrane domain, and then the cytoplasmic domain. So it has three domains. So the growth hormone receptor, they have two subunits, and there is a binding site on each subunit. So when the growth hormone binds to one receptor subunit, the binding site on another subunit is attracted, and both the subunit, they come closer to each other. So this produces a dimerization of the growth hormone receptor, as it initiates the activation of a number of enzymic cascades, including the JAK-STAT pathway of hormone action. So this receptor neither resembles any of the G protein linked receptor, nor it is similar to the receptor with the intrinsic tyrosine kinase activity. Mechanism of action of the growth hormone. Dimerization is essential for the receptor activation, as it initiates the activation of the number of enzyme cascades, including the JAK-STAT pathway. So the initial changes in the phosphorylation pattern of the cytoplasmic and the nuclear proteins is very important. It also stimulates the transcription of specific genes, including that of the insulin-like growth factor 1, prolactin, and other growth factors also act through the JAK-STAT pathway. Mechanism of action of growth hormone. The binding of the growth hormone with the growth hormone receptor activates 4 signaling pathways. So let us discuss one by one. JAK-STAT pathway. So there is JAK-STAT pathway. So JAK2 is a membrane of the family of cytoplasmic tyrosine kinases, which is also known as Janus kinase. It is associated but not the integral part of the growth hormone receptor. STAT, it is signal transducers and activators of transcription, which belongs to a family of inactive cytoplasmic transcription factor. So the binding of the growth hormone with the growth hormone receptor activates the cytoplasmic domain of it, which induces the JAK kinase. So the STAT on phosphorylation by the JAK kinases, it migrates to the nucleus. This results in the induction of various genes that mediates the hormone action. The next one is HHCGRB2. So the JAK2 phosphorylates the intracellular protein called as HHC, which in turn activates the GRB2 protein. So this GRB2 protein stimulates the MAP kinases that induces the various gene transcription in the nucleus. Next is the phospholipase C. So the hormone receptor complex also stimulates the membrane enzyme called as phospholipase C, which results in the production of DAG, so diacylglycerol. So this DAG initiates calcium influx into the cell, and probably also that initiates the calcium initiates the gene transcription. And then IRS, the activation of the JAK tyrosine kinase causes the phosphorylation of the insulin receptor substrate, which in turn that induces various intracellular enzymes. So these are the four signaling pathways which helps in the mechanism of action of the growth hormone. Physiological effects of growth hormone. Growth hormone is a anabolic hormone with profound short-term metabolic effects and also long-term growth promoting effects. So the acute metabolic effects are like lipolysis in the adipose tissue, decreased glucose uptake by the muscle, and stimulation of gluconeogenesis by the hepatocytes. So these are due to its direct actions on target tissues. So long-term effects are mainly mediated by the somatomedins. So that is the insulin-like growth factor 1, direct actions of growth hormone, effects on the growth. The most striking and specific effect of growth hormone is the stimulation of linear growth, which occurs due to its action on the epiphyseal cartilage on the long bones. The growth hormone stimulates all aspects of metabolism of the chondrocytes, the cartilage forming cells. So the major action of the growth hormone on the chondrocytes include the incorporation of proline into the collagen and its conversion into the hydroxyproline, and then incorporation of sulfate into the proteoglycan chondroitin. So this hydroxyproline and chondroitin together they form the extracellular matrix of the cartilage. So the growth hormone promotes cartilage matrix formation. There will be increased amino acid uptake and protein synthesis and increased RNA and DNA synthesis in the chondrocytes. There will be increase in the size and the number of the chondrocytes. Growth hormone also acts directly on the progenitor or the stem cells like prechondrocytes in the growth plates of the bone. So it facilitates the differentiation of prechondrocytes to the chondrocytes. So all these actions they result in the growth of epiphyseal cartilage, which helps to increase the length of the bone and subsequently that promotes the linear growth of the body. Thus the growth hormone increases the height of the individual before the closure of the epiphysis. Actions of growth hormone. So on the bone, growth hormone stimulates the activity of the bone modeling units. So it increases the activity of both the osteoclasts and the osteoblasts. However, the osteoblastic activity predominates and therefore the bone formation becomes more. So the total bone mass and the mineral content of the bone obviously increases. On the skeletal muscle, the growth hormone increases the growth of the skeletal muscle by increasing the protein synthesis and causing the hypertrophy of the skeletal muscle cell. It also promotes the activity of the satellite cells of the skeletal muscle. These satellite cells are the progenitor cells present in the skeletal muscle. Hyperplasia and hypertrophy of the visceral organs. So the organs like liver, kidney, heart, pancreas, intestine, and many endocrine glands, skin, connective tissues, they all increase in size and the function. So these organs grow in size due to the protein synthesis increases, and there will be increase in the RNA and DNA synthesis. So there will be increase in the cell size and number. So the general visceral functions like digestion or cardiac output and the GFR, they all improve. Pubertal and gonadal growth. So the growth hormone increases the height during the puberty. It also sensitizes the gonads to the actions of the luteinizing hormone and follicular stimulating hormone. So the growth hormone promotes the prepubertal sexual maturation. Effects on protein growth. The effects of the growth hormone on general somatic growth are primarily due to the protein anabolic effects. They include the positive nitrogen and phosphorus balance, and there will be increased amino acid entry into the cells in many tissues, increased RNA and DNA synthesis in the cell. There will be increased protein synthesis, and it also decreases the plasma level of amino acids and urea, which occurs secondary to the amino acid entry into the cell, and that increases the protein synthesis. Effect on carbohydrate and fat metabolism. So growth hormone is a pro-diabetogenic hormone because it causes the hyperglycemia by its action. It increases the release of glucose from the liver by facilitating the hepatic gluconeogenesis, and then it decreases the glucose uptake by the skeletal muscle. It also decreases the insulin sensitivity. So growth hormone stimulates the insulin secretion indirectly by increasing the ability of the pancreas to various insulinogenic stimuli. So there is insulin-like growth factor 1 mainly secreted from the liver under the influence of the growth hormone, which has the insulin-like activity. So indirectly growth hormone also promotes growth, as well as insulin is a growth stimulating hormone. Effects on the fat metabolism. So growth hormone causes lipolysis and increases the plasma level of free fatty acids. By increasing the free fatty acids, it promotes the ketogenesis. As it increases the free fatty acids and keto acids in plasma, growth hormone provides energy in various conditions of hypoglycemia and the stress. Effect on electrolyte metabolism. Growth hormone influences the concentration of various electrolytes by its own mechanisms. Growth hormone increases the plasma calcium by increasing its GIT absorption, and also potentiates the effect of vitamin D. It also causes sodium retention by decreasing the sodium and potassium excretion in the urine. It maintains the extracellular fluid volume indirectly by stimulating the RAS system and suppressing the action of the atrial natriuretic peptide on kidney. So growth hormone decreases the sodium and the water excretion. It increases the plasma phosphate level by increasing its reabsorption from the proximal tubules of the kidney. Effects that is mediated through the insulin-like growth factor. The effects of growth hormone on growth, cartilage, and protein metabolism depend partly on the production of insulin-like growth factor from the kidney, which is also called as somatomedinC. They are found to mediate the somatic effect of the growth hormone. So the principal circulating somatomedins or IGF-I, also called as somatomedinC, and insulin-like growth factor 2. So 1 and 2. So this 1 and IGF-1 and 2 have similar actions as insulin, and that is why it is called as insulin-like growth factors. So they are mainly synthesized in the liver. They are also formed in the cartilages and other tissues. IGF-1, it is more potent in mediating the effects of growth hormone than the IGF-2. So it is secreted independent of growth hormone before birth itself. However, after birth, this IGF secretion and actions are mainly dependent on the growth hormone. And it rises in childhood and attains its peak during puberty and declines in old age. It binds with the plasma protein called insulin-like growth factor binding proteins. There are 6 various such binding proteins, so which serves as a reservoir for the insulin-like growth factor in the plasma. And then insulin-like growth factor 2, so it is independent of the growth hormone action and plays an important role in the fetal growth. So this IGF-2 receptor is a mannose-6 phosphate receptor. In adults, it expresses only in the choroid plexus and meninges. It plays a main important role in the fetal growth alone. So the plasma concentration is around 300 to 800 nanogram per milliliter. So the comparison of the IGF-1 and 2. So 1 is mainly found in the liver, 2 is from various cells, but in adults it is mainly found in the choroid plexus and meninges. So it is regulated by the IGF-1 is regulated by the growth hormone after birth, and also nutritional status plays an important role, whereas IGF-2, so regulation is not clearly known. So the plasma levels of IGF-1 peaks at puberty. So the major physiological role of IGF-1 is on the skeletal and the cartilage growth, whereas IGF-2 plays its role mainly in the fetal growth. Catch-up growth, it is following a illness or starvation during childhood, there is a period of catch-up growth. So this takes place during which the growth rate is greater than the normal. So you can see this period. So the accelerated growth usually continues until the previous growth curve is reached. then is slows down and it normally it moves. So the mechanism behind the catch-up growth is not clear. Applied aspects of the growth hormone: hypersecretion of the growth hormone in adults. So after the fusion of the epiphysis, it results in acromegaly, and in children before the fusion of the epiphysis, it results in the gigantism. So this is the main two difference, the acromegaly and the gigantism. So acromegaly, it means the enlargement of the acral parts, that is the peripheral parts. So due to the tumor of the somatoscopes of the anterior pituitary, usually it is associated with proliferation of lactotropes also. So prolactin secretion is increased in these patients, around 20 to 40% of the patients. It is also due to the extra pituitary causes like increased hypothalamic secretion of the GHRH. Here you can see the acromegaly. Acromegaly, the characteristic features are enlargement of the acral parts of the body, especially hands and feet. Prognathism, it is the protrusion of the lower jaw. Glucose intolerance, acromegalic faces, overgrowth of the malar frontal and the facial bones. Hirsutism, it is increased amount of the body hair and hypertrophy of the soft tissues. Osteoarthritis due to skeletal changes. Gynecomastia and lactation. Increased heel pad thickness. There will be visual field changes, that is bitemporal hemianopia, which causes headache. Visceromegaly, increase in the size and functions of the viscera, and also macroglossia. Skeletal deformities occurs as bones enlarge in width. Bones cannot grow in length as epiphyses are already closed. Diagnosed by typical clinical presentation or computer tomography scan or MRI scan, and also by the high level of the growth hormone in the plasma. Surgical removal of the tumor is the best option to cure the disease. Here you can see the facial features of the patient with acromegaly. You can see the facial lines here. You can see the forehead bulge over here, facial lines, and prognathism. So it is the protrusion of the lower jaw, you can see, and here swelling of the hands, right? So how enlarged it is, the acral parts, the periphery. So here you can see the invasive growth hormone secreting macroadenoma. So lifting the optic chiasma, so which causes a bitemporal hemianopia, so which extends to the paracellular region. Gigantism, it is the hypersecretion of growth hormone before the fusion of the epiphysis. So pituitary tumor during childhood or hypothalamic during tumor causing the excess of the GHRH secretion can cause the gigantism. So it is before the closure of the epiphysis. So they become abnormally tall with gigantic stature. So other features are same as acromegaly except the acromegalic phases. So disease is diagnosed typically by the clinical presentation or the imaging by imaging the pituitary and the hypothalamus, and also by the plasma level of the growth hormone. So the earliest cure is the surgical removal of the tumor, which secretes excess growth hormone. Hyposecretion of the growth hormone, deficiency of the growth hormone, it results in the dwarfism. So this may occur due to four various causes. So first is due to growth hormone deficiency, hypothalamic failure, growth hormone secretion is decreased, that is the pituitary failure, decreased insulin-like growth hormone synthesis by the liver as seen in the African mice, and decreased or unresponsive growth hormone receptors. So that they are called as the Laron dwarfism. So when the growth hormone receptors are unresponsive, so all these things can cause dwarfism. Pituitary dwarf, it is due to the growth hormone deficiency. It is characterized by the short stature and correspondingly delayed bone and sexual maturation, decreased muscle and total body mass, decreased cardiac function and decreased bone density, they are all common. Mental power is relatively normal in a pituitary dwarf. This helps in differentiating them from a thyroid dwarf, wherein thyroid dwarf, in whom the mental retardation is a outstanding feature. It is diagnosed by the low level of growth hormone or insulin-like growth factor in the plasma. Pituitary dwarfism is treated by the administration of the growth hormone preparations. So due to the difference in the structure, there is wide species variations in the growth hormone. So growth hormone which are extracted from the animals, they will not work in the humans. So the amount of growth hormone collected from the human pituitaries at the time of autopsy is not adequate to treat the pituitary dwarf patients. So presently there are recombinant DNA technology there is. So by that technology, the large quantities of growth hormones are produced, and it is used to treat the pituitary dwarf patients. In summary, the growth hormone acts on the adipose tissue and causes lipolysis, which releases the free fatty acids, and it also acts on the carbohydrate metabolism, causes the decreased glucose utilization as it is a pro-diabetogenic hormone. It acts on the liver and it synthesizes the insulin-like growth factor 1. So this IGF-I acts on the cartilage and bone and increases its growth, and also acts on the muscles and other organs, which causes the amino acid synthesis. Books to read: Hindu Purana, Guyton and Hall, Genong, and G.K. Paul. Thank you very much for your active attention.